Hello, everyone. My name is Cassandra Brickert, and I have been working in Dr. Spiritu's lab for the last few years of pharmacy school. My capstone project is on the synthesis, folding, and purification of what are called 591 conotoxins. Specifically, I worked with conotoxins M3B, CnIIIE, and CnIIIF. So what are conotoxins? Conotoxins are peptides that are derived from cone snail venom. You may have heard of a medication called ziconotide, which is actually an FDA-approved conotoxin that is administered intrathecally for the treatment of severe chronic pain. This is an example of a conotoxin being used for pharmacologic treatment. The conotoxins that I worked with are known as 591 conotoxins. This represents their intercysteine loop size, or more simply, the number of amino acids that are between each of their cysteines. If we remember back to our biochemistry, cysteines have the ability to form disulfide bonds. The way disulfide bonds are connected and the intercysteine loop size I mentioned before are really important for the activity of conotoxins and are highly conserved in pharmacologic families of these compounds. The conotoxins I worked with, M3B, CnIIIE, and CnIIIF, each have 6 cysteines within their sequence. This allows them to form several different isomers. However, for the purposes of our project, we are only interested in producing one specific active isomer. Now, you may be asking yourself, why are we producing these conotoxins in general? In a preliminary study conducted by Dr. Espiritu and John Taylor, M3B was found to inhibit potassium voltage-gated ion channels, specifically channels 1.1 and 1.3. These are particularly interesting because they are found to be overexpressed in patients with disease states such as multiple sclerosis. This created interest in what potential activity these compounds have not only at potassium ion channels, but a wide variety of ion channels such as sodium channels. Thus, my goals for this project were to synthesize, fold, and purify these conotoxins in order to be able to screen them at a wide variety of ion channels. We will actually not be conducting the testing at the ion channels. We have a collaborator in Belgium that will receive our final product and run the assay for us, giving us information about what kind of activity these compounds have at receptors. One of the challenges that I was overcoming in this project was trying to minimize loss of product while optimizing purity of the final product itself. You will see throughout my results that we trialed different methods for synthesis and used different strategies to identify problem areas within our synthesis. So for my methods, my process always started with constructing these peptides and began with the synthesis process. This was done using solid phase synthesis, which was conducted within an automated synthesizer. If you look to figure 1, you will see a diagram outlining solid phase peptide synthesis. In short, we start with a resin. There are several different kinds of resin, but we used a rink amide and a clear acid resin. In my poster, you will see specifically acid resin synthesis data. The resin is like a bead that has lots of different linker regions on it, where amino acids can be added to form our peptide. Our resin always had a starter amino acid on it with a protecting group. On the image, you see BOC or BOC on the end of the compound. We actually used one called FMOC, but the role of this is to keep the amine from reacting until we want it to in order to form the correct sequence of amino acids. The deprotection step you see there is to remove those protecting groups. The appropriate amino acid is then added to our resin. We then have the coupling step, where the amino acid reacts to form a peptide bond, which is starting the formation of our peptide. After the coupling step, there is several what we call washes, where a solvent is used to wash the resin and peptide to remove any unreacted reagent and amino acids. This can interfere and form impurities within the peptide. This repeats over and over again for each amino acid. In the case of my peptides, there is 22 amino acids, so this occurs 22 times. For those that are curious, I added a photo of our automated synthesizer. It's down in figure 2. Some of you may be familiar with doing this synthesis process by hand, but this equipment actually does all of those steps for us, which is very helpful. It takes about 12 hours to synthesize these peptides. What we did have to do by hand was the remainder of the steps, specifically the cleaving process. Cleaving is essentially just removing the peptide from the resin so that we would be left with just our newly created peptide. We specifically used a reagent L cleaving strategy. After cleaving, we would need to take our sequence and fold it into its 3D structure so that our peptide would be in its conformational shape and have activity at receptors. For our peptide with all those cysteines, this meant the formation of disulfide bonds that I was talking about earlier. Once the peptide is folded, the last two steps are desalting, which is exactly what it sounds like. It is removing salt that remains mixed in with our peptide from the folding process and the mixing of our folding buffer. This has to be removed in order for us to isolate our peptide. This was done with reverse phase HPLC with a constant 5% gradient, which allows us to wash the salt off and then collect our peptide from the column. Unfortunately, this is where I identified a lot of loss of our product, and I will actually talk about that a little bit later. The last step is purification, which is isolating our specific isomer that we are looking for and collecting it with a purity greater than 95%, which is optimal. What this looks like is determining our isomer peak from the HPLC and collecting that specific peak for the synthesized peptide. Once those steps are completed, we have our final product. Yay! And then we can send it to our collaborators for ion channel testing. So for my results, they are separated by each one of the peptides I was mentioning before. So my first section is M3B. Below that is CnIIIE. And on the right side, you'll see the data for CnIIIF. Each of these compounds, I included a mass spectrotrace of what is called the crude peptide, which is just the peptide directly after the synthesis and cleavage process. You are able to see these in figure 2A, figure 3, and figure 4B. These confirm that we have created our intended peptide by the confirmation of expected masses that the peptides would have when sent through a mass spectrometer. Accompanying those mass spectra are the results of our HPLC traces. I have also provided sequences. For example, you can see in the figure 2A inlay 2 that show how our peptide would look after synthesis, as long as everything has gone to plan. It is in its reduced state. Going back to biochem again, this is when the sulfur of our cysteine carries a hydrogen instead of having our formed disulfide bonds. However, once we form our peptide into its 3D conformational shape, we get the formation of the disulfide bonds, and this is represented in the figure 2B inlay. What figure 2B is showing is that what our peptide looks like once we have folded it. This HPLC run looks very different than the one above, which has our crude peptide. Folding the peptide changes its charge and polarity, which means that it interacts with the HPLC column very differently than the crude one. On figure 2B, you can see several different peaks. Our hope is that that tallest, strongest peak is the isomer of our interest. In this case, it was. But each of those peaks actually represents each different isomer that could have formed during the folding process. The way we confirm that the isomer of our interest is that tallest peak is we collect each one of those peaks and then we run them through the mass spec again to see if it contains our expected oxidized folded masses. If you look down to figure 3, this shows the peptide CnIIIE. The biggest thing I want to point out is that if you look at the HPLC inlay 1, the trace looks very messy, unlike the trace for M3B. There is no strong, obvious dominant peak, which means that this compound did not synthesize cleanly and our product has several impurities. There is still a lot of work to be done with this compound, and what we learned is we need to explore why the conotoxin is harder to produce and how we can form a cleaner product. If you look to figure 4A and B, this is showing the crude form of peptide CnIIIF, which is the last peptide that I synthesized. The mass spectra in figure 4A is actually a separate synthesis from figure 4B. In figure 4A, we use a synthesis technique known as end capping. This can be done after the coupling stage that I spoke about earlier to stop any unreacted amino acid from reacting with our peptide. This helps prevent any deletion products from forming, which are a form of impurity. This, however, it can also cause lower yield of our product. We did this to hopefully better synthesize CnIIIF in figure 4A compared to figure 4B. With capping, we actually did not produce our intended masses, and it made our product less pure. It also cut our yield in half. After doing byproduct analysis, so this was essentially looking back at the other products other than our intended product, to see what those sequences looked like. We ended up not finding that we had very many deletion products. Instead, we actually had a lot of addition products. So instead, we tried increasing our washes after the coupling stage, which did help improve our synthesis. In figure 4C, you can see our folded CnIIIF product, which is going to be a similar kind of trace as the M3B figure 2B. So for conclusions, I discovered that increasing the washes was able to increase our yield and purity of our products, specifically for CnIIIF. For end capping, it is important. I learned that it is important to assess your byproducts to see whether you have deletion products as your main impurity, to assess whether this would be a useful tool for your next synthesis. One of our biggest problems was the loss of product during synthesis. I had mentioned earlier that we had observed that the desalting and purification process were the steps with the biggest amount of loss. We ended up modifying the HPLC to allow a larger volume or sample size volume. Thus, instead of doing several really, really small injections where the product could be lost, we were able to do fewer, larger injections, which seemed to help load our peptide onto the column and improve how much product we were losing. For the future, as I had mentioned, our CnIIIE is still in early stages, so we need to figure out what we need to do to improve that synthesis process. How this looks is we want to do a byproduct analysis to assess what kind of impurities we are forming to see what we can do in the synthesis process to negate that. We also want to assess our cleaving strategy to make sure our sequence of amino acids is compatible with the method we are using to cleave. And then we also want to try differential protecting strategies for all of our peptides. This will help better direct the folding of our peptides into that correct isomer that we want, instead of having several different isomers like the peaks I was showing you on our HPLC. That is all I have for my presentation. A special thanks for Dr. Sputu for all the help over the past couple of years and working with me in his lab. Thank you for listening to my presentation.